So in this lecture, we will see the different analysis procedures for water for bacteriological pollution. Okay, so we'll see the bacteriological tests. Before moving on to that, we have to know what is bacteria and why we are conducting a test for bacteria. So there are many microorganisms in water. There are many types of microorganisms. All natural waters except from deep boreholes contain bacteria. Actually, it is exposed to what is that? Atmosphere. It is exposed to land. It is exposed to human beings. So it may contain many types of bacteria or many types of microorganisms. Main source of bacteria is from soil, vegetation, life and matter on earth's surface. And bacteria is a single cell organism. It has no nucleus, no chlorophyll. It will multiply by binary fission for various sizes and shapes. So this is the peculiarity of bacteria. Its length is 1 to 4 microns, very small. We cannot see it with our naked eyes. And its presence indicates oxygen demand. It will use more oxygen and there will be an oxygen demand in the water where bacteria is present. And viruses they are smaller than bacteria and grown in medium to count the colonies or culture. So this is the peculiarity of bacteria and viruses. So we will see the classification of bacteria. Bacteria are of mainly two basic classification are there. One is based on harmness. Based on the harmness it is caused, and based on the oxygen demand. There are two types. So based on harmness there are of three types: non-pathogenic, pathogenic, combined group of pathogenic and non-pathogenic. So pathogenic, you all know, they are bacteria which causes diseases. They are harmful to human beings. They can cause typhoid, cholera, hepatitis, etc. And non-pathogenic means they are not harmful to human beings. They are sometimes useful also. And combined group of pathogenic and non-pathogenic they are present in intestines. Okay, intestines of warm-blooded animals, in human intestines and all. Some pathogenic, some bacteria are there. They are both useful as well as causes some diseases. So Bacillus coli and Bacillus bacterium, which is present in intestine, is one of the example for that kind of bacteria. They can be used as a representative organism or indicator organism. So we'll see what are indicator organisms later. Now based on oxygen demand there are three types: aerobic, anaerobic and facultative. Aerobic means it flourishes in presence of oxygen. It will multiply in number. Anaerobic means it can flourish even in absence of oxygen. Even without oxygen also it can multiply in number. And facultative means it exists in anaerobic condition also but cannot multiply. It can multiply only in aerobic, but it can exist in anaerobic and aerobic condition. That kind of bacteria are known as facultative. So there are two types, based on harm and based on demand of oxygen. Now these are some facts about bacteria. So there are certain things which is peculiar to bacteria. Some varieties are cultivated under lab conditions, some cannot be cultured. So in lab we can cultivate bacteria for some purposes, some testing purposes or some culturing, some analysis and all. We can cultivate bacteria in some lab conditions. Many of the bacteria form colonies of yellow, brown, red, violet or green colors. So they can form colonies. We can count the colonies by means of this color. Okay. Only a few varieties of bacteria can tolerate both acidity or alkalinity. Some continues to live and multiply, other don't. Some can continue in number, some can multiply, but other cannot. It has an optimum temperature for multiplication. Each type of bacteria has an optimum temperature for multiplication. Bacterial purity varies with sources: river, stream, lake, shallow well, deep well, etc. According to the sources, whether there is bacteria is present or not, how much amount of bacteria is present and all varies. And the density of bacteria in water depends on the quantity and quality of food available for them in water. Okay, so bacteria need some kind of food, right? So according to the food availability in water, the quantity and quality of bacteria increases, or it depends. Nutritional requirements are less and hence can multiply even in deep well waters. They need less nutrition, so they can multiply even in deep well waters where there is no nutritional content. And seasonal changes will affect the multiplication of bacteria, of course. Amount and remoteness of sewage and manual pollution have great concern in bacteriological pollution of water. Of course, the sewage and manures which we are disposing to water can cause bacterial multiplication. Presence of gaseous constituents, organic matter, inorganic matter in water can affect the bacterial multiplication. Metals like copper, silver and aluminum has detrimental effect on bacteria. Bacteria cannot live in such water which contains metals like copper, silver and aluminum. It is not ion, it is on. Sedimentation is a potent factor in bacterial pollution of water. Of course, sedimentation sometimes causes the increase in bacterial multiplication. Okay, these are some of the facts which is concerned with bacteria. Now we will move on to the indicator organism. How can we find the presence of bacteria in water? There are some indicator organisms. So indicators are some organisms which can be easily detectable in water by means of some chemical tests. Okay, so there are many kinds of bacteria which are growing in water, but we cannot find out all the types of bacteria in the water because there are no such chemical tests or found out procedures for finding such kinds of bacteria or viruses or some other organisms. But there are certain things, certain bacteria or certain microorganisms which can be easily found out by some simple chemical tests. Such kind of bacteria can be used as an indicator for finding the presence of bacterial pollution. Okay, that kind of bacteria are known as indicator organisms. Escherichia coli or E. coli or coliform groups are some of such bacteria which can be used as an indicator organisms. That means the presence, if such kind of bacteria are present in water, we can say that the water is polluted with some other bacteria also. The water is bacteriologically polluted, we can say like that. So E. coli, you all know, it lives in human intestine. Its presence indicate recent contamination with fecal matter or disinfection by superchlorination is the only solution. So fecal contamination of water, contamination from septic waste is the indication of E. coli presence in bacteria, in water. And coliform group, all the aerobic and facultative anaerobic bacteria, gram-positive non-spore forming rod shape that ferment lactose with a gas formation with 48 hours at 35 degree. If the water is kept at 35 degrees Celsius for 48 hours, coliform group can be analyzed. The presence of coliform group can be analyzed. And gram-positive means the culture gives red stain. When it is kept at 35 degree for 48 hours, it will see if it is giving red color, then it is known as gram negative, and if it is giving blue color, then it is given as gram positive. Okay, so each type of bacteria has its own indication. Now there are many tests, many objectives of bacteriological analysis. We are doing the bacteriological analysis of water for achieving certain objectives. So these are certain objectives: to find out the sewage or manual pollution, to ascertain the efficiency of chlorination. So after chlorination, if you are doing the test, we can ascertain the effectiveness of chlorination, how effectively the chlorine disinfects the water, that we can analyze. To find out the change in quality of water, to study the degree of pollution in river waters, to study the pollution of sea water, to work out the efficiency of purification systems. After filtration and all, we can analyze the bacteriological pollution of water. So that will give the effect of filters or efficiency of filter. To work out the pollution by bathers of swimming pool water. So all these are the objectives of bacteriological analysis of water. Now we will move on in detail about the bacteriological tests. So there are mainly two types of tests. One is membrane filtration or MF test, and the other one is most probable number test, MPN test. They can be of two types: total count or agar plate count test and B. coli test. So there are two kinds of bacteriological tests. One is membrane filtration. The name itself, from the name itself you can get that membrane filtration. So the water is filtrated by means of some membranes which can retain bacteria. Okay, so this is known as membrane filtration technique. And most probable number test is a chemical test which uses some chemicals for the identification of bacteria. So there are two subcategories, total count or B. coli test. We will see each of them. Okay. So this is the difference between MPN and MF test. MPN is actually, it requires some 48 hours for a positive result. It will take more time. Okay. Just like our COVID test, PCR test will take some 3 days and antigen test will get the result within minutes. So that is the difference. Okay. So, but it is more, it will take more time and it is more labor intensive. It is membrane filtration is less labor intensive because filtration is a simple procedure, but the analysis is a very large procedure, and it requires more culture medium. It requires more glassware, more test tubes are needed, more conical flasks are needed. It is more sensitive. MPN test is more sensitive to chemical analysis. We can if the chemical if some chemicals are if some quantity of chemical is changed, some quantities, particular quantities is changed, it will affect the test. Okay, that is the thing. And the result obtained indirectly by statistical approximation. Even though it is precise, it has low precision because it is an indirect result. We are not just counting the bacteria. We are getting an indirect result by means of a chemical test. So it is less precise, and it is not readily adaptable for use in field. We cannot use this procedure in field. It is a lab test. MPN is a lab test. It is a chemical analysis, and it can be applicable to all kinds of water. It is consumables readily available in most. So its cost is uh, less when using chemicals. In case of membrane filtration, it is just opposite to this. Okay, so that is the difference. Now we will see what is membrane filtration. As I have told earlier, there is a membrane which can retain the bacteria. We will pass the sample of water through this membrane, and the membrane has a capacity to retain the bacteria. Okay, the indicator organisms are retained on the filter, then transferred to a suitable selective culture medium in a petri dish. That retained bacteria or retained organisms are transferred to a petri dish, and that petri dish is incubated. That petri dish containing this organism is incubated. Okay. Now, after the incubation, we can visually identify the colonies of bacteria formed. The colonies of bacteria. Bacteria will not be occurring in a single unit. They will be occurring as colonies. Okay, a number of bacteria will be grouped into one colony. Okay, so they can be seen as colonies. If they are gram negative, we can see red colored colonies there. So 1, 2, 3, 4, like that we can count the colonies. So there will be colony forming units, CFU. So how can we detect this? We will count the colonies per 100 milliliter of original sample. So if we are taking 100 milliliter of original sample, we are filtering the sample, and we will get some filtrate. Some organisms we will get as filtrate. These organisms are transferred to a petri dish. It is incubated, and after that we will count the number of bacteria. So that is the CFU per 100 milliliter of original sample. So like that we will get. It is a direct result. And the second test is most probable number analysis. It is a statistical method based on the random dispersion of microorganisms per unit volume in a given sample. And some color change is seen. So we will see in detail about this MPN test. The MPN tests are mainly of two types: total count or Agar plate count test and B. coli test. So the B. coli test is again has three steps: presumptive, confirmed and completed test. Okay, so we will see each of these tests, total count and B. coli we will see. The procedure of total count or Agar plate count test is given here. First what we will do is, the bacteria are cultivated in specially prepared medium of agar with different dilutions of sample of water with sterilized water. Okay, what we are doing is, we will take the sample water. Okay, we will take, we have to analyze the water from a river. Okay, for example. So we will take a sample of water from the river. Okay, and we will dilute this water with sterilized water. Okay, we will dilute this water with sterilized water. The dilutions may be different. For example, first we will take 10 ml of water and we will dilute we will mix it with 10 ml of sterilized water. And another sample we will take 10 ml of water and we will mix it with 100 ml of sterilized water. Like that, different dilutions we will take. Okay. And these dilutions are incubated for 24 hours at 37 degrees Celsius. Okay. They are known as hot counts, or 48 hours at 20 degrees Celsius. They are known as cold count. Either of the way you can incubate it. Either you can incubate at 37 degrees Celsius for 24 hours or 20 degrees Celsius for 48 hours. You can give hot counts or cold counts. Now, if bacteria is present, they will compulsorily form colonies. For sure they will form colonies. Then the bacterial colonies are counted and the results are computed for 1 cubic centimeter. The CFU per milliliter is equal to colony forming unit, number of colony forming unit found here, into the dilution factor. Dilution factor means, I have already told you that the sample of water is diluted in a different way. Different dilutions we will take. For example, 10 milliliter of sample with 10 milliliter of sterilized water means 1 dilution factor is 1. If 10 milliliter of sample is diluted with 100 ml of water means dilution factor is 0.01. Like that, dilution factor into 1 by amount of sample, that is aliquot. So if you are taking 10 milliliter of sample means here aliquot is 10 milliliter. So that will give the CFU per milliliter. So this is the procedure. We are just taking the sample, diluting it with sterilized water. Different dilutions we will take, incubating the diluted water. You can do two types of incubation. One is hot count and the other one is cold count. Either of the incubation you will do. After that, surely bacteria will be formed. We will count the bacteria. The CFU into dilution factor into 1 by aliquot will give the CFU per milliliter. So this is the total count or agar plate count test. Now we will move on to the second test. It is B. coli test. So this is the most important test that we are using today. Okay, B. coli test. It has actually 3 steps. One is presumptive test, second one is confirmed test, and the third one is completed test. Presumptive test is based on the ability of the coliform group to ferment the lactose broth and producing gas. Presumptive test actually, it gives the ability of the bacteria to ferment the lactose broth. A broth will be there, lactose broth, a solution will be there. And if the bacteria, this coliform bacteria has the ability to ferment this broth. So this capacity is taken as presumptive, as the basis for presumptive test. Confirmed test consists of growing bacteria on media which suppress the growth of other organisms. And the completed test is based on the ability of the culture to again ferment lactose broth. So there are 3 steps: presumptive, confirmed and completed. We will see each of them. So first one is the presumptive test. This is the steps for presumptive test. I have already told you that the basis of this test is the ability of the bacteria to ferment lactose broth. Okay, so first we will take definite amounts of diluted samples of water. For example, 0.1 cc, 1 cc, 10 cc, etc. So these are the dilutions. Okay, we will dilute the water in different amounts of sterilized water. Now this water is placed in standard fermentation tube containing lactose broth. There are fermentation tubes containing lactose broth. These samples are taken and placed in that fermentation tubes containing lactose broth. It is incubated at 37 degrees Celsius for 48 hours. After this period, if gas is produced from the tube, then we can say that the test is positive and E. coli bacteria is present in it. Otherwise, the test is negative. Negative test tells us that the water is fit for drinking. This is about presumptive test. We are diluting the sample. We are placing this sample in a fermentation tube containing lactose broth, incubating it at 37 degrees Celsius for 48 hours. If gas is produced after this time, the test is positive. E. coli is present. Otherwise, test is negative. If test is negative, we can use the water for drinking. Otherwise, we have to do confirmed test. Okay. The next step, we'll move on to the next step otherwise. So confirmed test can be done by two means. Two procedures are there. Here, this is the first procedure and the second procedure will be given in the next slide. In the first procedure, what we are doing is brilliant green lactose bile is taken in a fermentation tube. This is a chemical. Brilliant green lactose bile is a chemical which is available in all the chemical labs. This bile is taken. A small portion of the lactose broth showing positive presumptive test is carefully transferred to yeast. So earlier we have done presumptive test. Some test shows positive result. That that test which showed positive result is taken. That lactose broth which showed positive result is carefully taken and placed in this brilliant green lactose bile. Okay, and it is kept for 48 hours. If gas is seen in tube, then the test is positive, otherwise the test is negative. If the test is positive, we have to do the completed test. Okay, that is the thing. This is the first procedure for confirmed test. Another procedure is also there. In this, plates containing endo or eosin methylene blue agar, it is another chemical. A plate containing this chemical is taken. As earlier, this lactose broth which is showing positive presumptive test is marked on this plate. Just it is marked on this plate. And it is kept for 37 degrees Celsius for 28 hours. If colonies of bacteria are seen, how can we identify the colonies? It will show some dark spot or metallic brightness color. If it is showing some color like this, then the test is positive, otherwise the test is negative. If the test is positive, we have to do the completed test. Okay. So if all these things, this is the confirmed test. If all these things is positive, then we have to move on to the completed test. This is the last step of B. coli test. Okay. So in this completed test, we will introduce or inoculate the bacterial colonies into lactose broth, fermentation tubes and agar tubes. So the test positive from the confirmed test, the lactose broth or whatever it is from the confirmed test or presumptive test is taken and it is inoculated again at 37 degrees Celsius for 24 to 48 hours. Okay, maximum of 48 hours it is inoculated. If gas is again seen, then we can say that the completed test is positive, otherwise the test is negative. If positive, we have to do detailed test for finding the type of bacteria present. So we have confirmed that there is bacteria present. Now we have to do whether which kind of bacteria is present in that particular water. So this is about completed test. So these are the different tests that have to be conducted for the bacteriological analysis of water. Now there is a term known as B. coli index, which we have to note. It is an index or number which represents the approximate number of bacteria per cc of sample of water. Per cubic centimeter of water, how many number of bacterial colonies are present? That is known as B. coli index or B. coli number. So a procedure for that B. coli, calculating the B. coli index is there. First we all know that we will conduct the presumptive test with the different dilutions of sample water with sterilized water. We will do a number of tests. With using the same sample, we will do a number of tests. Okay. Out of that, some will be positive and some will be negative. From the same sample, some will be positive and some will be negative. Because when we are taking samples, it may not be always representative. Okay, some samples may be pure, some samples may be of bacteria, maybe containing bacteria and all. So a number of tests is conducted and some may be positive and some may be negative. So we will calculate the percentage of positive result. So if we are taking one dilution of sample and we are conducting 100 tests by using the same sample, out of that 100, 50 are positive and 50 are negative. So the percentage of positive result is 50%. Okay, half of that is positive, so 50% is positive. Now we will take the difference between the successive positive percentage. So we will see it using a calculation. Here you can see, we will first take the proportion. We will take the dilution. First 10 cc is taken. 10 cubic centimeter of water is diluted with some sterilized water. So this is the first dilution. We will do 100 test using the same sample. 100 presumptive test using the same sample. Same procedure will be followed. Out of that, percentage of positive result is only 1. Out of 100, only 1 is positive. So 1% is the positive result. And using 1 centimeter cube sample, we will again do the 100 test, and out of that 0.7% is the only positive result. So we got these two, and in the next step we will find the difference between these two, difference between the positive results, successive positive results we will find. And in the next step we will find the reciprocal of column 2. What is column 2? Column 2 is the dilution. Okay, we will find the reciprocal of this dilution. 10. Reciprocal of 10 is 0.10. Reciprocal is 1 is 1 itself. So we'll find that this. In the final step, we will multiply these two quantities. The difference into reciprocal of column 2. We will multiply. And we have done this test for many dilutions. We have done the same test for 5 dilutions of sample. So we will get 4 results. So we are summing up all these results. We will get 5.68. So this should be less than 10. This this is known as Becoli index. This 5.68 is known as Becoli index. This should be always be less than 10 for potable water. So that is the thing. So this is about today's lecture. Thank you.